The last topic for this lecture will be the prokaryotic cell, and that is intentional. We are not going to be talking about the eukaryotic cell, as eukaryotic cells are covered in quite a bit of detail in general biology. So anything in the chapter that covers eukaryotic cells is not going to be covered here, and those things that are similar between eukaryotes and prokaryotes also will not be covered here. So feel free to ignore that information and focus instead on what we're talking about in this presentation. So as you can see here, the prokaryotic cell, though it lacks a nucleus, which is a defining characteristic of a prokaryote, is not at all simplistic. It contains many extracellular appendages as well as lots of things going on inside. Typically, prokaryotic cells are surrounded by a cell wall, as are fungi and plants, which is exterior to the plasma membrane, which you'll remember from general biology is the barrier between the cell and the outside environment. Obviously lacks a nucleus there, but does contain a nucleoid region where the DNA is highly coiled, and though it doesn't show it in this picture, is actually attached to the inside of the plasma membrane for increased stability. There are also the equivalent of the membrane-bound organelles you find in eukaryotic cells, and those are called inclusions here. So it's not that a prokaryotic cell lacks organelles, it's that prokaryotic cells lack membrane-bound organelles, which really includes the nucleus when you think about it. So to say there's no organelles is kind of a misnomer. We don't call them organelles, but if we're talking about a sequestered space within the cell in which a particular job is done, then certainly the inclusions would work in that capacity. So technically speaking, it's just that a prokaryote has no membrane-bound organelles. So we're going to take a tour through the cell. We're going to start outside, move inside, and end up in the middle there talking about DNA by the end of this lesson. So without further ado, let's talk about prokaryotic cells. It turns out the first thing that you need to talk about when you talk about prokaryotic cells is shape. Because most of them contain a cell wall, most prokaryotes also have a rigid, fixed morphology or shape. And those 6 shapes are shown at the top of your screen there. Round cells are referred to as cocci, which in its singular form is coccus, which I know is a little weird. But these are Latin words, and in Latin the S is actually singular and I is plural. So cocci, or a coccus, is a round cell. A cocobacillus is sort of a rounded rod. A vibrio is a curved rod. A bacillus is actually the technical name for a rod-shaped cell. And then at the bottom there, you see 2 kind of curvy cells. One is called a spirillum, and the other is a spirochete. So there, as you can see, are multiple kinds of morphologies. We focus in general biology on the cocci and bacilli. Don't talk a lot about these other morphologies, but it is important to know that they exist. And when we talk about cocobacilli or vibrio or spirilla or spirochetes, we always talk about them in singular arrangement. They're not found attached to one another. So if you look at the right-hand side of your screen, you see a drawing of a vibrio and then a scanning electron micrograph of some vibrio cells. You can see a bit of a spirillum there and then a spirochete, and really look there between the spirilla and the spirochete and notice the difference in how coiled those cells are. And that's one big difference between those two. When they're drawn, they have a tendency to look very similar, but in reality, the spirilla are typically just curved a little bit, whereas the spirochete are very tightly wound. And we're going to find out in a few minutes that in addition to that, spirochetes have very different ways of moving. So hang on for that, and then let's just move over to the cocci and bacilli, which is where we spend most of our attention. Before we do that, though, I forgot to mention the cocobacilli, which are rounded rods. And the reason we don't really talk about those in general biology very often is, as you can see, they're very small. Notice the 1 micron bar next to that picture, that scanning electron micrograph. So these are incredibly small, and so small really that with our microscopes we can't make out the difference between cocobacilli and cocci, and most of the time if you were to see those under the microscope you would just assume they're cocci. So again, let's focus on the cocci and bacilli. So both of these cells, like the Vibrio and the cocobacilli, the Spirilla, and the spirochetes, can exist as single cells. And what that means for all of these cells is once they undergo binary fission, which is a topic that we will talk about later on, but it's the way that bacteria reproduce. You kind of think of it like the prokaryotic equivalent of mitosis and cytokinesis. Some cells, when they undergo that process of reproduction, will divide completely and break apart, and that is always what happens for the spirochetes, Spirilla, Vibrio, and cocobacilli, and it is sometimes what happens with bacilli and cocci. So they will divide, break off completely, and remain as separate entities. However, some cells are programmed to undergo complete reproduction. So they're completely separate cell membrane, separate cell wall, but they remain attached to one another. And when they do that, they become diplococci or diplobacilli. And if they are also genetically programmed to keep that connection for extended periods of time, those diplobacilli will undergo binary fission and grow into chains, which we refer to as streptobacilli or streptococci. As I mentioned earlier when we were talking about naming conventions, remember there's a genus Streptococcus who is named that because of the streptococcal arrangement and morphology of the cells when they are seen under the microscope. So streptococci are chains of round-shaped cells, and streptobacilli are chains of rod-shaped cells. Now, that sort of arrangement exists when a cell undergoes binary fission on a single plane of division, and that's the only kind of division that bacilli can undergo. So these are the only possibilities for bacilli. The cocci, on the other hand, because they are radially symmetrical, can undergo division on more than one plane. So you see here in the middle of that diagram, cells that are undergoing division on 2 planes can grow into a tetrad arrangement, and cells can actually undergo binary fission on 3 planes, which will allow them to grow into clusters, which are shown at the bottom here and are referred to as staphylo arrangements. So we have streptococci, which are arranged in chains, and we have staphylococci, which are arranged in clusters. And that name, staphylococci, may sound familiar to you because we mentioned a genus of bacteria earlier on, Staphylococcus, with a species named aureus. Staph aureus is named Staphylococcus aureus because those cells under the microscope have a staphylococcal morphology and arrangement. So now that we've got the morphology and arrangement down, we're ready to move into the cell. To begin our discussion of the prokaryotic cell, as I mentioned, we're going to start from the outside in. So initially, we're going to be talking about the parts of the cell which exist outside of the cell membrane and so are external to the cell itself. So there are 4 of these that we generally talk about. And there is a summary table there that will give you some information about each of these. The first of the external structures that we're going to talk about is something called a glycocalyx, and a glycocalyx is a polysaccharide that is thought to be excreted by all bacterial cells. Difficult to get evidence for that because it's a polysaccharide. It's very difficult to keep it intact during staining processes so that we can see it under the microscope, but for those cells that secrete enough of this polysaccharide material, they will either arrange it in a very structured form, in which case we call it a capsule, and that's shown here in turquoise around the dark cell. If that glycocalyx is less organized and just sort of oozes out from the cell, it's called a slime layer. So capsules and slime layers are both kinds of glycocalyces. Both of them are used by cells to attach to a substrate. So regardless of whether you're talking about a human pathogen or an environmental bacterium, they can attach to a substrate in their environment, which is kind of their preferred way of doing things. The capsule especially is very helpful for pathogens because it prevents phagocytosis by white blood cells. And we're going to be talking about those capsules later on when we talk about the difference between two strains of Streptococcus pneumoniae, one of which is encapsulated and is deadly to mice, and the other, which is not encapsulated, which can be very quickly cleared by the mouse's immune system. So the capsule is highly pathogenic. The slime layer is more of an environmental thing with environmental bacteria. Both of those, though, glycocalyces. Next up, we have the pilus, which is shown here, whose plural form is pili. Pili are long, slender. Notice the micron bar at the bottom of the diagram here. Long, slender, hollow tubes through which DNA can be transferred from one organism to another, and that's a topic that we're going to pick up on later in the course. Fimbriae, which are hair-like projections on the surface of the cell and are commonly mistaken for cilia. They're not cilia. They have nothing to do with motility. They don't move. They're rigid structures. The closest thing that you could think about fimbria being like would be Velcro. So they're really helpful for bacteria to attach to a substrate. Like the slime layer, they help their attachment. And that can increase their pathogenicity because if they're able to attach to a surface and your body defenses are trying to get them out of the body, this will increase their ability to cause disease. So fimbria, or fimbriae in the plural form, are small hair-like projections that work like Velcro. And finally, the flagella, which, like the flagella of eukaryotes, are used for motility, although the flagella are very different in their structure and arrangement, as you can see here, and the way that they are utilized by bacteria. They are very long, very thin projections from the cell, as you can see from that 0.8 micron bar at the bottom there. And so we typically can't see them under light microscopy, and so you see here a transmission electron micrograph in order to see those flagella. So of these four external structures, we're going to revisit some topics later on in the class, but today we're going to focus on the flagella and talk about how the prokaryotic flagellum is unique from the eukaryotic flagellum. One thing that's unique about prokaryotic flagella is the way that they are arranged on the cell. So typically, eukaryotes have a single flagellum, and the most probably common example of that would be the human sperm cell, which has one single tail, and it whips that tail and kind of swims like a tadpole would through the water. And that is a possibility for microorganisms. As you can see in the second picture here, we have a cell with a single long flagellum. That flagellum is oriented at the pole or end of the cell, and so we call that a polar flagellum. We also refer to that flagellar arrangement as monotrichous, and monotrichous is Greek for single hair. So trichous means hair, mono of course means one. So monotrichous flagella describe an arrangement where a single flagellum is oriented at the pole of the cell. So if we go all the way over to the left, we have peritrichous, which again has Greek roots, which may seem kind of frustrating to a 21st century student, but you got to remember when people were learning about microorganisms and naming things, it was common for people to interact with one another in Greek and in Latin. Those were the scholarly languages. So I don't think it was, I oftentimes think of people just trying to make things overly complicated. It actually would have been very easy for an earlier student to understand what that meant, because they would know that peri means around and trichus means hair. And so if you said peritrichus to them, they would automatically picturing something that has hair all over the surface of it. And you might recognize peri as a prefix in the word perimeter, right? So the perimeter is the outside edge of an area, and peritrichous flagella cover the outside edge of the bacterial cell. So we have peritrichous, monotrichous, lophotrichous. Lopho means tuft. So you can see there are several flagella in a group at one end of the cell. So lophotrichous is a tuft of flagella at one end of the cell, and amphitrichous flagella are on either end or either pole of the cell. And that word amphi may sound familiar to you because we have amphitheaters, right? And in amphitheaters, the audience is in the front and on the sides of the stage. So amphi means both sides. So amphitrichous flagella are on both sides of the cell. So that's different. The other thing that's different about these flagella is that they are rigid structures. They do not whip like the eukaryotic flagella, they spin. And typically they spin in a counterclockwise rotation, and that allows the cell to swim through the liquid environment that it's living in, and randomly the cells will change the direction of that rotation, and that will allow them to change the direction in which they are swimming. When you have a polar flagellum, it's all fine. You swim forward when it's in counterclockwise, and when it goes in a clockwise rotation, you swim backward. When you have flagella all over the surface of the cell and you change the direction of those flagella all at once, what it will do is it will cause the cell to tumble. So the tumbling of the cell is a result of the changing of direction of the flagella. So that tumbling that we saw earlier in the lecture, where van Leeuwenhoek was noting the spinning movement of the cells, those were flagellated cells that were changing the direction of their flagella. So we see what we call runs in bacterial swimming, and then we see tumbles as they change direction. And those things happen randomly just as the bacterium is surveying its environment. However, motility in bacteria, like in eukaryotes, is a way of responding to environmental changes. So motility is not always going to be a random running and tumbling. We're going to see in just a moment that these runs and tumbles are intentional and they're directional so that the bacteria can get to places it likes and get away from places that it doesn't like. Before we do that, though, we want to talk about one more unique thing about bacterial flagella, and that is that some bacteria, spirochetes, have endoflagella, flagella that are inside of the cell. Those are also sometimes called axial filaments, and they anchor at one end of the cell, and they turn in a counterclockwise direction and change direction to clockwise just like other flagella, but because they are inside of the cell, the whole cell twists and turns around the flagellum. And the really cool thing about spirochetes is that when you see them move, you're actually seeing the flagellum move because the cell is just changing its shape around the flagellum. So these are special, and these exist in spirochetes, and that's one of the ways that you can tell the difference between the spirochete and the spirilla, is that you can see in this scanning electron micrograph the axial filament running the length of the cell. So that's one more interesting, unique thing about these guys. Now, let's talk about why bacteria have flagella, right? Because we want to keep in mind whenever we're talking about things that cells have, and this will be more meaningful to us once we talk about metabolism and the way that cells produce ATP to fuel cellular processes. A flagellum is a huge structure made up of many, many proteins, and so it's energetically expensive. So it's not going to be there unless it provides a necessary job for the cell that it's attached to, and it turns out it's highly useful to the cell. And the cells that have had that flagellum studied the most are members of the enteric bacteria, which is a Greek word that means intestine. So intestinal bacteria who have peritrichous flagella have been studied in detail for their ability to move, which is called taxis. So enteric peritrichous taxis is what we're going to talk about here. And it carries over into a lot of bacteria. Just this is the specific example that we're looking at. So I'm not going to ask you what kind of taxis did we look at. This is just kind of for your information. So taxis means movement, right? So think taxi. You get in a taxi when you need to go from one place to another and you don't have your car. Taxis is movement. And the first thing that we want to understand about bacterial taxis is that these teeny, teeny, teeny little guys sense gradients in a very different way than we do. We are large enough that we can sense gradients spatially, meaning one half of your body can feel warmer than the other half of your body. And so you, if you're feeling too warm, can move your whole body into where you were feeling cooler. And if you're feeling too cool, you can move your whole body into the area that felt warmer. And that works because you're big, right? And when you're small, you can't sense gradients spatially. You have to sense them temporally, which means if you're cold, you just move and see if you're warmer now. And if you're warmer now, then that's good and you stay there. But if you move and it's still cold, then you move some more. And then if it's warm enough, you stay there. So what you do is you move toward, right? So you move randomly. But then when you get somewhere that is the environment that you like, you try to stay in that environment. So you move towards something. And the easiest way, I think, for humans to understand how this works is to consider our sense of smell, because our sense of smell involves chemicals that are in the air and we breathe them into our nose and they hit mechanoreceptors in our nose, and those receptors cause a chemical change that sends a nervous impulse into the brain that the brain registers and reacts to. And that's just the way that bacteria sense things in their environment. They have receptors which bind chemicals from the environment, and they decide whether or not those chemicals are something they like or something they don't like. And then they respond to that with swimming or taxis. So think about yourself blindfolded, walking into a room that you are not familiar with, and you smell chocolate chip cookies. And I say to you, find the chocolate chip cookies using nothing but your nose. So you start walking, and as you're walking, you're smelling, and you know that you're heading in the right direction when the smell of the cookies gets stronger. And so you keep walking and you keep walking and you keep walking, and the smell is getting stronger and stronger and stronger. And so you keep walking in that direction. And then you take a step and you notice that the cookies smell has decreased. So what do? Do you keep walking in that direction? No, you change direction and you try to get back on the trail of those chocolate chip cookies, right? So you are moving as the concentration of the cookie smell gets stronger, you keep moving toward it, and every time the concentration gets lower, you move away from it. And that's exactly what bacteria do. As they move randomly, when they find a higher concentration of something they like, they stop tumbling and they run in that direction. And if they move past that increased concentration and the concentration of what they like, the attractant, has started to go down, they tumble so that they can go back into that area. So they change direction, trying to get back into the high concentration. Now, go back to that thought experiment. You're walking, right? Your sense of smell is heightened. You've got a blindfold over your eyes, and all of a sudden you get a big whiff of rotten egg, right? Do you keep walking toward the rotten egg? No, you turn and you go away, right? And it's the same thing with bacteria. When they swim towards something that stinks, right? That's a repellent that they don't want, they don't like, they tumble. They tumble and they get away from there as fast as they can. So just as you're thinking about these guys, just think about yourself. Think about yourself moving toward chocolate chip cookies, moving away from rotten eggs, and that will do a really good job of helping you to understand how these guys move in the environment. And the way that they do that is, granted, a very complicated process, and I don't want you to get hung up on the details. What I want you to understand is these little red circles are chemicals in the environment, And whether that chemical is something they want, like food, or something they don't like a waste product, they will bind that, right? So it will bind to a receptor on the surface of the cell, and that will cause what some of you may remember as a signal transduction pathway that will ultimately flip the switch on the flagellum and change its direction and cause that tumble. So you can imagine every time a molecule hits that, that starts that chain reaction and changes the level of tumbling and running. So we don't need to get wrapped up in all of these details. What I want you to do is understand there's some environmental stimulus, and that affects the way that the flagellum turns. And interestingly enough, that stimulus can be a chemical, right? So there is chemotaxis, there is positive chemotaxis toward a chemical that you like. There is negative chemotaxis away from chemicals you don't like. There are also receptors on the surface of some cells that absorb light. They absorb particular wavelengths of light, and that particular wavelength of light may be something that the cell wants and moves toward, or something the cell doesn't want and moves away from. And it's really interesting. Bacteria that are able to undergo photosynthesis will not just move toward light. They will move toward particular wavelengths of light that they photosynthesize best in, and they will exhibit negative phototaxis and move away from wavelengths of light that they don't photosynthesize in. So it's highly sophisticated, not just, oh, that seems cool, I think I'll go there. It's a very highly specialized receptor that's interacting with that flagellum and changing its direction. So it's not a random thing. Motility is one of the basic ways that these cells respond to environmental changes. All right, so that leads us to the cell wall. We're still outside technically because the plasma membrane is the barrier to the cell, but let's talk about that cell wall and then we'll be ready to get inside. And the cell wall of bacteria is a very important structure because prokaryotes are all unicellular. And as a unicellular organism, it can't regulate the environment of its cells, right? Your body can regulate the environment of all of your cells with the exception of your skin cells, which are exposed to the outside environment. If you're a single-celled organism, you have nothing, like you're not big enough to do anything about the external environment. So really what that means functionally is that you're always living in an extreme environment, whether it's hot or cold or salty or not salty, or there's waste products, or there's not enough food, or all of these things are constantly changing. And those changes, one of the big things that those changes do is changes the way water wants to equilibrate. And most of the time what that means is that water wants to get into the cell because the concentration of solutes is much, much higher inside the cell than outside the cell. And with that pressure comes the likelihood that the cell will burst from osmotic pressure, which is called osmotic lysis, right? So I put a picture of a balloon blowing up here, right? Because if water goes in, and boom, the cell blows up. And one thing that can prevent the cell from doing that is the cell wall. So that's a general use of a cell wall. You probably talked about that in general biology in reference to plant cells, which also have cell walls to prevent osmotic lysis. And actually the plant cell wall helps the plant to stand up tall, right? That's how you know when your flowers and your plants need water, is they start to wilt, they start to sag, and when you give them water, they stand back up again. So cell walls prevent osmotic lysis, and cell walls are not unique to prokaryotes. The reason that we're talking about the cell wall here is that the prokaryotic cell wall in its structure is unique. So not in its function, but in its structure. If we think about cell walls generally, there are a couple of different kinds of organisms that have cell walls. Fungi have cell walls, which are made of chitin. Plants have cell walls that are made of cellulose. Archaeans, as you can see here, have cell walls that are made of something called pseudomurian. And bacteria have cell walls that are made of something called peptidoglycan. So it doesn't mean that all cells, all prokaryotic cells have cell walls. As you can see there, it says usually peptidoglycan, but then right under that there is a caveat. There are something called mycoplasmas, which lack cell walls. So it's very common to have a cell wall. When the cell wall is there, it's composed of peptidoglycan. When you're talking about bacteria, if you're talking about archaeans, if the cell wall is there, it's made of something called pseudomurian. So archaeans have pseudomurian, plants have cellulose, fungi have chitin. We're going to focus on the cell wall of bacteria, which is made of peptidoglycan, which, as its name suggests, is made up of both proteins and carbohydrates, right? Peptido referring to the protein portion, glycan referring to the carbohydrate portion. When we think about this peptidoglycan cell wall, the best way to sort of picture it is to think of a brick wall where all of the bricks are made up of the carbohydrate portion and all of the mortar or the cement between the bricks is made up of the peptides. So it's highly organized, very rigid, very strong, and all cells that are genetically programmed to make this peptidoglycan cell wall have a particular amount of peptidoglycan in their cell wall. So you go all the way from having no cell wall, like a mycoplasma, to cells like those members of genus Staphylococcus who have a tremendous amount of peptidoglycan in their cell wall. And then everybody ranges in between there. So everybody has a genetically predetermined amount of peptidoglycan, right? The thickness of the brick wall that they will make outside of their plasma membrane. And that, it turns out, is the basis of the work that Hans Christian Graham did. Remember, we said that he had no idea that what he did was going to revolutionize microbiology. He, in fact, was trying to find a stain that would differentiate prokaryotes from eukaryotes. But what he did, without knowing it, was stumble on a technique that would tell you how much of this molecule, peptidoglycan, was in that bacterial cell wall. And the reason for that is that a cell with a lot of peptidoglycan, as is shown here, will trap the primary stain of the Gram stain procedure and hold on to it even when the cell is decolorized. And so what a gram-positive cell actually is a bacterial cell with a ton of peptidoglycan. So when we say gram-positive, what we're really saying is that's a cell that has a lot of peptidoglycan and retains that purple stain in the Gram stain procedure. So on the left-hand side of your screen, we have both a scanning electron and a transmission electron micrograph of that cell wall. So in the larger transmission electron micrograph, you can see that thick layer of highly organized peptidoglycan right outside the plasma membrane. Incidentally, you can also see a daughter cell that is attached to this bacillus. And so you can see there where that diplobacilli arrangement comes in, where you have two cells that are absolutely separate from one another, two distinct plasma membranes, two distinct cell walls, and yet stuck together in this diplo arrangement. So this peptidoglycan, you can see from the inset, this scanning electron micrograph creates a very smooth structure on the surface of the cell because it's, as I mentioned, highly organized, very strong. Now that might lead you to believe that gram-positive cells are more capable of withstanding environmental stressors than gram-negative cells, but it actually turns out that that's the opposite. That's something we're going to be talking about in a later unit, is the fact that gram-positive cells actually have a harder time surviving in the environment than gram-negative cells. And the reason for that will come clear in just a moment. But before we move on to that, I just want to reiterate, a gram-positive cell has a thick layer of peptidoglycan. The reason these cells are called gram-positive is because that thick peptidoglycan layer allows them to retain the primary stain, crystal violet, and stay purple throughout the staining process. Now, let's look at the alternative, very little peptidoglycan. When a cell has very little peptidoglycan, It is stained purple during the gram stain procedure, but then during the destaining procedure, that cell is destained, becomes decolorized, and then has to be counterstained with this pink stain, safranin, that is not washed out of the cell. And the reason that works is because that very thin layer of peptidoglycan can't retain the primary stain. So I was mentioning that this might lead you to think that the cell would not be able to survive in the presence of environmental stressors. And the way that these gram-negative cells get around that is that outside of that thin layer of peptidoglycan, they have something called an outer membrane. And that outer membrane is highly resistant to chemicals and other things like that. It's a lipid bilayer membrane, but it's embedded with a lot of polysaccharides. So unlike the phospholipid bilayer membrane of all cells, this outer membrane is called a lipopolysaccharide outer membrane, and it's constitutionally different. It's constructed differently than that cytoplasmic membrane, and it turns out that the way that that lipopolysaccharide outer membrane is put together makes that cell highly resistant and allows gram-negative cells typically to survive chemical disinfection and things like that much better than gram-positive cells. Again, if we look at the transmission electron micrograph of this cell, we can see that very thin layer of peptidoglycan, and we can see outside of that this outer membrane. Now, as a membrane, it's not very highly organized, and you can see the difference there in the inset scanning electron micrograph. The surface of the gram-negative cell doesn't look smooth and nice like the surface of the gram-positive. It's kind of wavy and disheveled looking, and that's because of the presence of that outer membrane. So when Hans Christian Gram stumbled across this procedure, he didn't just give us a way of identifying gram-positive and gram-negative cells as a way of categorizing them. It turns out that there are antibiotics that target peptidoglycan and target the outer membrane and things like that. And so knowing whether a cell, an infectious organism, is gram-negative or gram-positive allows us to begin treatment that is more focused. So not just against bacteria, but against gram-negative bacteria or against gram-positive bacteria. So it really was revolutionary, and it's too bad that Gram was not alive to understand just how amazing a contribution his gram stain technique was to the field of microbiology. And I bet you thought this next slide was going to be inside the cell, but we have one more thing to talk about before we get there, and that is a very special kind of cell called an acid-fast bacterium, who contains a great deal of peptidoglycan. That's kind of a bummer in the way that this diagram is made, because there is a thick layer of peptidoglycan. So these guys will stain gram-positive, but exterior to that thick layer of peptidoglycan is a layer of something called mycolic acid, which is a waxy lipid that makes this cell highly resistant to environmental chemicals and disinfectants and things like that. It also makes this cell really difficult to stain. So even though they do stain gram-positive, it's hard for either of the stains to get inside them. So under a gram stain, what you'll typically see is kind of a streaky purple color that has areas of white inside it, and that is indicative of acid-fast bacteria, because it's really hard for any stain to penetrate that waxy outer layer of the cell wall. Now, there is a special staining technique, which is called the acid-fast staining technique, where heat is employed to melt that waxy layer, allowing stain to penetrate the cell. And in that procedure, the heat allows the stain to penetrate the cell. The cell is then allowed to cool, and then it is decolorized with an acid alcohol. And only those cells with mycolic acid are capable of holding on to that stain through this acid destaining process. And so we call them acid-fast cells. So cells that have mycolic acid are acid-fast cells. Cells that do not are non-acid-fast cells. And there are gram-positive and gram-negative non-acid-fast cells, because the defining characteristic that makes the acid-fast stain differential is the presence of mycolic acid. So only those cells which contain mycolic acid will stain acid-fast during this staining technique. And if you look in the light micrograph picture, they're actually pictured in that pink color, and they're bacilli, but they kind of stick together because that waxy lipid, that mycolic acid on the outside of the cells, makes them adhere to one another. The non-acid fast cells are those round blue cells, those blue staphylococcal cells that do not contain mycolic acid, and so could not hold on to that pink color and had to be counter-stained with methylene blue. And this is one of the staining techniques that you're going to be looking at in lab. Now, this is not common. Having mycolic acid is not common. It's found in the genus Mycobacterium and also in the genus Nocardia. The most famous acid-fast bacterium is Mycobacterium tuberculosis, which of course is the causative agent of tuberculosis. So in a clinical setting, when you hear someone saying they're ruling out AFB or they're doing an AFB test, what they're probably talking about is a suspected case of tuberculosis in the hospital. The only other clinical option would be leprosy, which is far less serious, far less contagious, and is far less common. So in a hospital, when you hear AFB, you're probably talking to someone about tuberculosis. And of course, that's something that you want to diagnose very quickly, because not only is it highly contagious, it also is highly deadly. And there are lots of strains of tuberculosis today out there in the hospitals and in the community, which are multi-drug resistant and really hard to cure. So that's it. We finally finished the cell wall. So the cell wall, these are either thick peptidoglycan with or without this waxy lipid mycolic acid layer, or thin peptidoglycan with that outer membrane, or wall-less, right, as is the case with the mycoplasmas. And now we get to the plasma membrane. Right there, shown in yellow, the plasma membrane is the barrier of the cell to the external environment. Plasma membrane, just like the plasma membrane you talk about in any of your other classes, is a lipid membrane which contains receptor proteins and porins and all sorts of interesting things that allow the cell to interact with the external environment. It's semi-permeable. It allows some things in, keeps some things out, all of those things that you learn in general biology. But one thing it's not that you learned in general biology is structurally always a lipid bilayer membrane. So hold on to your hats. We've got to take a time out and talk about something unique about the plasma membrane in prokaryotes, and that is that the phospholipids of the archaeans is totally different than the phospholipid of every other organism on Earth. And I want you to take a minute to kind of to drink this in. So we have, there's some argument, but we used to have 5 kingdoms, right? We had kingdom Animalia, fungi, plantae, protista, and Monera. And Monera was a kingdom of all of the prokaryotes. And then people started studying prokaryotes, and they were like, whoa, wait a second, man. These monerans that you've grouped together are really diverse. There is hardly anything within this group that's the same, except for the fact that they don't have a nucleus. And really, we think that was a little bit hasty of you to give all of these kingdoms to a bunch of different eukaryotes and then throw all the prokaryotes into one kingdom. And that's where we got, instead of Monera, we got kingdom Eubacteria and Archaebacteria. And today, really, people don't even like the terms Archaebacteria and Eubacteria, because the archaeans are not bacteria. They are so different as to have their own group, completely separate from the bacteria. And that's something that we're going to be talking about very soon in class when we look at the diversity of microorganisms. So archaeans are prokaryotes, but the lipid membrane that surrounds archaeans is different from all bacteria, all plants, all animals, all fungi, and all protists. All of those other organisms, all eukaryotes and bacteria, have something called an ester linkage in their phospholipid membrane. Now, we're not going to get into the chemistry of that, right? But they're all the same. Archaeans are totally different. They have an ether linkage. So their phospholipids are totally different from everybody else's. In addition to that, the phospholipid of archaeans can either be a lipid bilayer orientation, like every other cell on the planet, or there are some archaeans who have a lipid monolayer. So at the top here, you have what's going to become part of a bilayer membrane, right? Because it only has one hydrophilic portion and one hydrophobic portion. So of course it has to have a little friend next to it whose hydrophobic portion will sidle right up with the blue area and whose hydrophilic green portion will be available to the aqueous environment of the cell interior, right? And then the other side will be happy with the aqueous environment of the exterior of the cell. At the bottom there, you have a monolayer, which has these two hydrophilic heads on either side, so it doesn't need to be oriented with a second lipid. It will orient into a perfect little membrane all by itself. So the function of the cell membrane is the same for every organism on the planet. The structure of the cell membrane is different for archaeans. That's the take-home point there. And there are two pictures here, scanning electron micrographs of bacteria. One's bacteria, one's archaea. Which is which? You have no idea, right? Because they look the same, and that's why they got grouped together, but it turns out that fundamentally and functionally they're very different from one another. All right, you ready to finally get inside the cell? Awesome, me too. Now, the organelles of the prokaryotic cell, which we call inclusions. We're going to talk about just a few common inclusions that can be found in prokaryotes. The list is too long to be exhaustive, so we're just going to pick a couple of them. The first of those is the metachromatic granule, which is pictured here, colored, which is where it gets its name. Metachromatic means you can color it. And this is a site of phosphate reserves, which may seem a little bit strange to you, but you got to remember that this is a single-celled organism cruising through the environment, right? And what do all cells need to make in order to reproduce? They need to make DNA, right? And what is the backbone of DNA made of? Phosphate. So if you come across a stockpile of phosphate, then you gather up all of the phosphate and hold onto it so that you can use it to make DNA and undergo binary fission and grow and make babies, right? Because that's how life goes on, is cells undergo binary fission and make other cells. So the metachromatic granule is a site of phosphate reserves. Cells also have a need for energy, and that energy comes in well-known to us forms like carbohydrates and fats, but it also comes to certain kinds of bacteria in a form that is completely foreign to us, and that is energy that is stored as sulfur, which sounds weird, right? I mean, you can get on board with the carbs, right? We eat, and our short-ish term storage is glycogen in our liver, right, which is a carbohydrate. So we understand having these large stores of carbohydrates. Of course, our long-term energy store is fat, so we can understand the storage of fat for long-term energy reserve. What is unique here is the idea that cells would store sulfur. And there are some cells, it turns out, who, in addition to being able to eat carbohydrates and fats, can actually eat sulfur, right? They consume sulfur and they use that to make ATP. So polysaccharides or carbohydrates, lipids or fats, and sulfur are different energy reserves and can be found in polysaccharide granules, lipid inclusions, and sulfur granules. Now, if you are a photosynthetic cell, you might compartmentalize the place where you perform parts of photosynthesis in something called a carboxysome, right? So carboxy for CO2 there. This is, you can kind of think about this as the prokaryotic equivalent of a chloroplast, perhaps. It's the site of the light independent portions of photosynthesis where CO2 is fixed or reduced or converted into a sugar, which will then be broken down for ATP by the cell to fuel its cellular processes. So the carboxysome is the site of carbon dioxide fixation, both in photosynthetic prokaryotes and also in some prokaryotes who undergo a process called chemosynthesis that we're going to talk about in a later lecture. So metachromatic granules, energy reserves, carboxysomes, gas vacuoles, which are kind of a fun inclusion that allows the cell to float. And floating is really important when you think about the fact that swimming takes energy and floating doesn't, right? So if you want to be able to float, let's say you're a photosynthetic cell and you want to be able to float on the surface of the water where the sun shines and allows you to photosynthesize, you don't want to have to be burning energy to swim there while you're trying to make energy via photosynthesis. So rather than swimming and actively trying to stay there, you passively float there, right? So that would be like the difference between you trying to tread water for 2 hours or floating in a life jacket for 2 hours, right? Obviously, one would take far less energy than the other. So gas vacuoles allow cells to be buoyant. And then finally, magnetosomes, whose name means magnetic body, are not there so that the cell can be magnetic. This isn't some kind of like navigational system or something like that, right? It is magnetic, so you can see there from the picture, the magnetosomes will line up along the, with the magnetic field of the Earth. But the reason for those magnetosomes and the reason that cells will collect and sequester iron oxide is to destroy hydrogen peroxide, which seems a little strange, I'm sure. But it turns out, and we'll learn about this in later units, that when you breathe in oxygen in order to use it, like you and I do, you will accidentally create hydrogen peroxide, and hydrogen peroxide is a poison. And we'll talk about why that is in later units. So every cell that breathes in oxygen has to have some way of dealing with hydrogen peroxide. You and I have catalase, as evidenced by the fact that when you put hydrogen peroxide on your cuts, it bubbles. These guys have iron oxide, so they don't have catalase, they have iron oxide, which allows them to live in an aerobic environment and breathe oxygen. So as you can see, a big variety in the kinds of inclusions, and I think other than connecting metachromatic with phosphate, energy with polysaccharides, lipids, and sulfurs, and so on and so forth, the thing to understand here is that the prokaryotic cell, though it lacks a nucleus, does not lack the sophisticated compartmentalization that's found inside eukaryotic cells. The difference here is that there are no membrane-bound organelles. These inclusions are not bound by a membrane, though the inclusion is an organelle in the sense that it is a sequestered area within the cell where a particular job is done. The nucleoid region, where we find a single circular piece of DNA, so that's different, right? You have linear pieces of DNA and you have 46 of them. Prokaryotes have one, and it's a single circular piece of DNA, but otherwise, as we're going to find in later units, is just like yours. And though it is not housed in a nucleus, it is highly packed to keep it safe into what's called a nucleoid region. And it is, though the picture here doesn't show you, attached typically to the inside surface of the plasma membrane to keep it from moving around inside the cell. Now, one totally different thing that prokaryotes have that eukaryotes lack is extrachromosomal DNA, which is also circular but much, much smaller, and that is a plasmid. So we're going to talk about that in much more detail later on. For right now, the hereditary material in the cell is double-stranded DNA, just like it is in every other cell, and there's a chromosome, and there are also plasmids. Before we wrap up this lecture, I just want to talk about one more topic from this chapter, and that is sporulation. So spores, you might remember from previous lecture, that some bacteria have the ability to form spores, and we can look for the presence of spores in cells using that differential stain, the spore stain, which is important for food safety, right? Because spores, it turns out, are highly, highly resistant to environmental stressors. So we talked about mycolic acid and how mycolic acid makes cells resistant to chemicals and other things. And we talked about the lipopolysaccharide outer membrane and how that makes gram-negative cells better able to withstand environmental stressors. And it turns out that of all of those things, the endospore is the thing that allows the cell to survive the greatest environmental stressors. And I'm saying endospore to differentiate the bacterial spore from the spores that you talk about with fungi that you probably talked about in general biology. So those are sexual spores. And the reason that fungi produce spores is to reproduce. Bacteria do not produce spores to reproduce. And you can get that from this picture very quickly. If you just move clockwise from number 1, you have a single cell, go all the way down to number 6, you have a single endospore, which is able to germinate into a single cell again. So if one cell becomes one spore becomes one cell, that's not reproduction. What the cell is doing this in response to is some environmental change which would end up killing this cell. And not all cells can form spores, and so not all cells can survive these terrible environmental stressors. But cells who do make these spores are capable of withstanding all kinds of crazy things. Like they've taken spores out into space, exposed them to the vacuum and radiation of space and brought them back, put them into broth, and they grow. They found, spores that are thousands and thousands of years old in Egyptian sarcophagi and put them into broth, and they've germinated and grown. So they allow cells to withstand long periods of time in a dormant state that is highly protected from environmental stressors and changes until the time is right, and then the cell can germinate. So the process of sporulation is only possible if you contain the recipe for how to make a spore. And of course, like all cellular recipe, the recipe is found in your genes. And the genes for sporulation are found in some cells. There are about 113 of them on average. And those genes, once you turn them on, the process of turning on those genes and beginning the process of sporulation takes about 8 to 10 hours to complete. And it starts with, as you can see from number 1 at the top here, the cell making a second copy, a new copy of its DNA, and then sequestering that DNA in a very small portion of the cytoplasm, right? So gets rid of a lot of the water, and it's very dehydrated, and then it starts surrounding the DNA in a membrane, and then it forms a septum around a second membrane, and then it fills that space between the membranes with peptidoglycan, and then a spore coat forms around the peptidoglycan membrane, and as the cell disintegrates and dies, the endospore is freed from the cell, and then that endospore cruises around for 5 minutes or 8,000 years or 12 years or however long, and then it germinates, and when it germinates, it grows back into a single vegetative cell that does life just like the cell always would do. So when you stain a preparation and are trying to stain spores, you have to do something to allow the stain to penetrate the spore. And typically we use heat to do that. And you really, really heat up these spores until you get that endospore coat liquid enough that the stain will penetrate it. And then you allow it to cool. And then the stain can't be washed out of it because the spore has encapsulated the stain, and you get a colored spore in your preparation, as we saw in the spore stain, and as you will see when you do your spore lab. Now, under routine staining conditions, such as in a gram stain, this cell would color either gram positive or perhaps not as likely gram-negative, but the spore itself would not be colored. So when you do a gram stain, just like you can kind of get a hint that you have cells that have mycolic acid and are acid-fast by the streaky purple that you see, when you do a gram stain, you can also get a hint at whether or not you have a spore former there, because you'll have a gram-positive cell which will have a clear oval or a clear circle inside of it. When you see that clear space, you think, some clear area inside of the cell was not able to be penetrated by the stain, either crystal violet or safranin, and that might be a spore, and then you do a spore stain to confirm that. So that's our last topic, the spore. And I want you to keep in mind, just like flagella, spores are not made by all bacterial cells. The ability to form a spore, the ability to make flagella are adaptations that some bacteria have and some do not.